food for other animals, muskrats, otters in some cases, raccoons, different fishes will feed on freshwater mussels. They also serve as shelter, just in the case of their shells being left behind. We see things like in the bottom right on my screen, you can see a little mad tom is taking shelter in an expired freshwater mussel shell. They are cavity nesters and they're little bitty catfish. They only get to be maybe 3 or 4 inches long, and they'll actually use shells or bottles or cans or logs or whatever to lay nests in, and then they can defend their nest. Freshwater mussel shells are often also they serve as places where fish can lay their eggs. If they're sitting on the bottom of the stream can flow underneath the shell and aerate eggs. These are probably darter eggs that are on the bottom of this shell here. So freshwater mussel shells are really important in the ecosystem. Freshwater mussels themselves, especially when there are a lot in a system, actually stabilize the stream bottom. So the act of the mussels burrowing in, they're holding tight to the substrate. What we think about mostly when we think about freshwater mussels is their ability to filter. Everyone says, you know, they're the livers of the rivers or they're indicators of good water quality. That's certainly true, but they're also improving water quality in places where they exist. They serve as primary filters, so they're actually filtering things out of the water. They'll store contaminants and things in their tissues and in their shells. They also do something called bioturbation, which is sort of, you can think about it like when you go and till up your garden in the springtime, you're turning over some of those things. So freshwater mussels, as they wiggle their foot down into the substrate, they'll actually be churning up some of those nutrients, make them available to other organisms. They'll help aerate the top section of the substrate. And they also perform deposition in the stream. So they're taking things out of the water column, different types of bacteria, algae, maybe some organic materials that they're processing, and then they spit it out, usually mixed with mucus that's a little bit heavier than what it was when it was floating in the water column. And other organisms like macroinvertebrates or benthic fishes can come along and actually feed on those sorts of things. So mussels are really beneficial to have in the watershed. And the other really cool thing about freshwater mussels, which I'm sure this group knows about, is that they have a parasitic life stage. So mussels have separate male and females. So the male releases sperm into the water, the female will filter it in and fertilize the eggs that she's holding. Those eggs will turn into larval mussels, which are called glaucidia. And then glaucidia get released whenever the time is right and hope to find a host fish. So for the most part, the mussels in the Fox River are generalists, so a lot of them will transform on many different fish host species. Some of them are specialists as well. They'll only transform on maybe darters or sculpin, and I'll talk more about that later. But all of the freshwater mussels that we have in the Fox River Basin have to have this parasitic life phase to get from being mussel larvae to being adult or juvenile mussels. So once they're on the fish, they might attach the gills or the fins, depending on the species. They'll stay on the fish for a month. Some species will overwinter on fish, but that's not as common. And then they will burst out of the little cysts that they formed on the fish, drop into the bottom of the river, and hopefully find a good place to live. So freshwater mussels are found all over the world. They occur on all continents except for Antarctica. We in North America, over here, we have over 300 species of freshwater mussels known. It was recently believed that we had the most species in the world, but some revisions to taxonomy has shown now that East Asia has more species than North America, but we're still doing really great with how many mussels are known worldwide. If we look at the upper central Midwest, that's what I made this map be. The diversity of freshwater mussels, we don't have 300 species here in the central Midwest, but we do have quite a few. So this map is showing you the Mississippi drainage here in blue. So all the major feeder rivers that go to the Mississippi River north of Missouri, we'll say, well, there's about 52 species known from this big drainage. If you go down to southeastern Illinois, there's about 82 species known that go to the Ohio River. These differences in numbers are mostly just due to the way the glaciers came through. The things that colonized this upper section, they came up the Mississippi River and spread throughout that watershed. The Ohio River drainage was able to draw from some of the species that were in the Cumberland Plateau that didn't get glaciated. It's a long story, but anyway, I just wanted to give you a picture of what diversity is in the upper Midwest so you can understand, like, is 20 species a lot or is 10 species a lot? So this is what historically is known from these drainages. Okay, the Fox River Basin is right here in this little yellow circle. So you know where we are in the world, which I'm sure you all are very familiar with the Fox River Watershed. It's a medium-sized river basin. So when we're thinking about the 52 species known from the Mississippi watershed, that's looking at the whole thing. So the larger the size of the watershed, the more animals are going to be present there. That's just how it is. So before colonization times, the Fox River watershed was an undulating prairie with woodlands and these kind of flowing wetland systems. Most of the watershed now is agriculture, and it's increasing in urbanization, especially in a few of these areas closer to Milwaukee and the suburbs of West Chicago. So what do we know about mussels in the Fox River? So we actually have a lot of historical studies, not a lot, we have some. There are historical studies that have looked at freshwater mussels in the Fox River, dating all the way back to the late 1800s, which is really cool to just have this data set. So because of the way the Fox River is situated and where it was close to Chicago, so close enough that people could come over on trains or horseback or whatever they were doing back in the late 1800s to collect samples of freshwater mussels. The historical studies that we have are not really comparable to what we're doing today. The records that we have are limited. A lot of times the location data is really limited. It might just say West Dundee. You don't know whether it's the river there or if it's a stream or if it's someone that had shells in a box that they got from their grandma and then they wanted to trade them. So we do the best we can with historical studies, but there are some limitations. So for example, our oldest study here from LaSalle County in 1874, that's just one county, that's what we know. Some of those collections were from the Fox, some of them were from the Illinois River. We're making some guesses. By and large, most of the information that we know about the Fox River watershed in terms of freshwater mussels focused only on the main stem river because it was the most important. The first really widespread tributary studies happened in the 70s. There was a study in that was published in 79 that basically printed an atlas of freshwater mussels for Wisconsin. So we have those data available. A few data sets from Max Matson in the 50s in Illinois, but only from a couple tributary watersheds. So for the most part, all the data that we have focused on the river until this important study in 2004 by Bob Schanzel and some others, and I'll be talking a lot more about this later. But I just wanted to highlight that was really our first real picture of freshwater mussels in the whole basin. So we know that there have been 32 species found in the Fox River. I'll say verified. There's a few shell records that are really dubious that, you know, may have been misinterpreted as the Fox River of the North, when really it's the Fox River of the Wabash system in Illinois. So 32 species that we believe were present in the Fox River. The Fox River itself was also the focus of this really interesting and depressing article written in 1914 by Mr. Eldridge, who was fisheries biologist, I believe, and it's about the pearl industry in the Fox River. So there's something about the Fox River that made really spectacular pearls and became a target for pearl hunters in the early 1900s, especially in the stretch, which is the lower stretch of the Fox main stem between Millington and Sheridan. And people were collecting mussels, cracking them open. They might have seen them to get the tissues to relax, but looking for pearls. And there were pearls of really extraordinary value for that time. So at the time of collection, they were valued between $1,000 and $1,800 for like an individual pearl, which in today's value is $30,000 to $53,000. And so this brought people out. This brought out tons and tons of pearl hunters to the area. For the most part, the shells were just discarded. They were looking for pearls. They didn't care about the mussel shells. They didn't necessarily even care about the tissues of the mussels, and they would discard the shells, but occasionally those got scavenged by other people for other purposes, and those were being sold off at, I think it was like between $3 and $7 a ton, which in today's dollar value is between $80 and $200 a ton for these mussel shells that have, are these long-lived animals. For the most part, the shells were gathered by wading, so people were just going out into the river. They weren't using the mule or the braille system like we see in all the pictures from most of the mussel button industry pictures. But I guess they did sometimes use the mule, and they would float them down, turn up the mussels, and then people were just out there, collecting all the mussels. So the most valuable species in this stretch of river was the mucket, which we actually don't see the mucket in the main stem of the lower river anymore. But just the sheer number of mussels that were present on the main stem Fox is like mind-boggling because we don't see that now. Then what they were estimating is around 1910, there would be 70 shellers per day on the river, up to 400 would go out on the weekends, which I think is incredible to think about because lots of times we don't see people recreating on the rivers. You might see a team of kayakers or a fisherman here and there, but you're not seeing 400 people out there on a Sunday afternoon. So it's just really interesting to think about. From these observations, we know that there were 12 species recorded in the main stem Fox River in Illinois in 1914. 4 of those species that were recorded in that stretch have never been recorded alive again in the main stem Fox River. I don't remember exactly which ones those are. I have them in a table if anybody's interested. But what you see from that time was a complete crash of the mussel fishery on the Fox River. So these are carloads of shells, and I'm assuming they're talking about carloads of like train carloads. And so in 1909, there were 11 carloads that were exported. So I think they were loading up all these discarded shells that then were being shipped to the mussel button factories. In 1910, you saw lots and lots of carloads of mussels that were coming out of the Fox River, and then it crashed. It completely crashed in 4 years. And then the main stem populations of the Fox River remain depleted to this day. We haven't ever seen anything like what was observed back in the early 1900s. Part of that, most of you may have seen this map that has been put together. This was from Santucci's paper many years ago. It's been adapted because some of these dams have come out, but there are a lot of main stem river dams on the Fox River. And so even if we had populations that had recovered on the main stem Fox, it would be difficult to get mussels back in these stretches. So that's my doom and gloom portion. I found the story fascinating when I was reading about the mussel fishery in the 1900s. I'm like, oh my gosh, it's hard to wrap your brain around. But in general, everywhere mussels are imperiled. Of the 300-ish species that we have in North America, we have lost a decent piece of the pie completely. Extinct means they are gone from the earth, not to ever come back. About a quarter of all the species in North America are endangered at the state or the federal level. Some of them are critically imperiled. They will probably go extinct within your lifetime, no matter how old you are. There are some that are hanging on by just a couple individuals that we have in hatcheries. Most of the species that we know about are at risk in some form. They might not be on a state list, but they're species in conservation need or that sort of thing. And then about 25% are considered stable. So these are your common species that are doing well. They're persisting in spite of all the alterations that we are seeing. So that said, we are interested in knowing more about freshwater mussels. We know that they have undergone rapid changes in the last 100 years. We've seen them go from really ridiculous abundance to the point that they never thought that they could over or outfish them, to having populations that are almost completely gone. The Illinois River itself went through the same kind of scene where mussel populations completely crashed, but we are now seeing a recovery in the Illinois River and some places like that. So we're seeing change, but it takes a really long time with freshwater mussels to measure change. Like I said, they need fish to reproduce, so you have to have a situation where the fish and the mussels are encountering each other. Freshwater mussels also burrow down into the bottom of the river. They're hard to find. Their juvenile stage usually stays kind of secluded for the first couple years of life. They might be in different habitats than we think of when we're looking for adult mussels. So it's hard to kind of map these changes. So I mentioned that study in 2004 that was published by Bob Schanzel. This project that we're working on is basically a repeat of that study. It's been about 25 years, which is a reasonable amount of time to see some changes in the watershed. And we need updated, reliable information to guide new conservation efforts. So in the turn of the century, around 2000, we were really just starting to think about freshwater mussels in terms of conservation. We've been sort of behind the times for freshwater mussels. We are still learning about them all the time. And so with that, we need some updated information. And then we also are interested in locating significant remaining mussel assemblages in the Fox River. And one other part of this project has been looking at the rainbow mussel. I'll talk more about rainbow in a little while, but part of the outcome is to augment rainbow populations. And through this project, we have been targeting populations of rainbow that we know exist to collect genetic samples, to look at the structure of all the populations that remain in the Midwest, and then hopefully be able to build those up. Okay, so these are the study sites that we've been looking at in this past few years of this study. So we have covered the entire watershed. We still have a few sites that we haven't been able to get to that are on our list. Hopefully this year will cooperate. So our preliminary results from what we've done so far is we've recorded 24 live species in the basin, which is actually really great. I think, I mean, if you think there were 32 known previously to the entire watershed and we're finding 24 of those alive, that's really remarkable. We collected 26 total species if you're including shells. So one of those is hickory nut, which was found only by shell alone at a couple sites in Wisconsin. We do not have the shell vouchers for those individuals that were collected. We just don't. So it's to be determined whether those were actually true IDs of hickory nut or not. This is one of those things where at the time, I don't know, you just make mistakes sometimes in the stream and we don't have the voucher. So Wisconsin's working on that to make sure if these were true shells of hickory nut that they would get passed on to the museum so that we can actually curate and catalog them and have them available to other scientists. Hickory nut is known from the watershed from a few shell records that have been saved at the Natural History Survey Museum as well as some other museums throughout the country. So it's not totally unreasonable. Either way, they're not believed to be present in the basin alive. Another species that we found only by shell is purple wartyback. It is a state threatened species in Illinois, and I believe it's state threatened in Wisconsin as well. It's known only from the main stem, to my knowledge, hasn't been collected since 1958 alive in the Fox River watershed. So we don't hold out much hope that purple wartyback would ever come back without some sort of intervention from humans. But either way, those are two species that we didn't collect alive. Everything else we found live individuals, which is really great. Also, almost all the sites that we surveyed had live mussels present. Certainly there are places in the Fox River watershed that don't have mussels, and we probably didn't go there because they didn't have mussels. The exceptions in our study of sites that we visited were headwater tributaries. So once you get too far upstream, if it's a stream that dries up over time, freshwater mussels don't really like that. So they don't really persist there, with some exceptions, sure. There was also one site on the Fox main stem in Wisconsin that no live individuals were recovered. It's a fairly impacted site. There have been mussels found there before, but this particular survey, we didn't find any. Okay, and I will also say that large sections of the Fox main stem in Illinois were not surveyed. So you'll see on our map kind of where we didn't go. The Fox in Illinois is difficult to survey. A lot of it is not wadeable. There's also boat traffic and anyway, so that said, we didn't go to a lot of places. There's always more work to do. Okay, so I'm going to go through a couple graphs here. Don't get too burdened with the data. I just wanted to show you all what the top 5, this is like the Family Feud results, top 5 most collected mussels in the Fox River. So this is looking at the entire watershed. This is what we were seeing. So among sites that had live mussels collected, these are our 5 most common species. These are the things that we will see at almost every single site: plain pocketbook, giant floater, white heel splitter, creeper, and ellipse. These are common species in the Fox Basin. This pattern will hold for all of the sites that we visited, is that these are species that we saw at most of our sites. I was also curious if they made up the most of the biomass or the most individuals collected. So we looked at that as well. The most abundant species or individuals, species with the most individuals, I will say, were ellipse. By and large, we found the most ellipse of any other mussel: plain pocketbooks, pimplebacks, muckets, and round pigtoe. We found over almost 7,000 individuals just in the study that we're looking at. 1,400 of those were ellipse. So ellipse are really prolific little buddies. Where they are present, they're usually doing quite well. Same with plain pocketbook. Plain pocketbook is a big mussel. They get to be like baseball sized, softball sized, and they grow quickly and they are really thriving in this watershed. So I'm going to show you a couple other graphs that look just like these. First I'm going to show you some pretty pictures. Sorry about that. These were the common species that we saw. Plain pocketbook here. This is that big old baseball. It's named cardium, which means heart. They're kind of shaped like a little heart. Pimpleback, very common. One thing that I have learned from looking at the historical data is that pimpleback were relatively rare in the early 1900s in any of the older surveys. So they didn't see pimpleback very often, and they're one of the more common species that we see now. So that's interesting, I guess. Giant floater, this is the Canada goose of the mussel world. They are everywhere. They don't really have specific habitat requirements. They usually live in slower moving water, but they can be in the swifter water. One of the sites that we surveyed near Algonquin on the main stem, there's a dam there, of course, but the river had cut a little side channel, but at that time in the summer, it had gotten cut off from the flow. So there was no flow in this little bitty like side pocket. And the giant floater that I found in that side pocket had floated to the surface. This is why they're named giant floater, is that when they're unhappy, they will fill up with air and pop out. And they wait at the top for higher water to come along and wash them out. So it was really cool to see with field crew and everything to be like, here's a giant floater, it's doing its thing. When they die, they'll also float, which is way grosser, but this one was alive and was just waiting for water to come along and wash it away into a better spot. So that's the giant floater. They are really, really variable. They can be long and darker colored, look more like this little guy down here, the creeper, or they can be this more kind of lake form that we see, which is a blown up jelly donut kind of mussel. White heel splitter, very common. They're big pancakes. They're very flat. All of these are pretty resistant to water quality issues. They can persist despite alterations. Creeper is this little brown one at the bottom. Creeper is one that is undergoing some taxonomic work. Kentaro Inoue at the Shed Aquarium, who's been working with us on this project as well, he's been looking at creeper populations and finding some really interesting things, that there might actually be some diversity beyond what we know now. So there may be additional species of creeper that's kind of tied up in the creeper that we know about. So these are our common species. The other one that's not pictured here is the ellipse, which I'll talk more about in a little bit. Okay, so when you look at the whole watershed, the Fox River watershed is very long. You're starting in Wisconsin, in southeastern Wisconsin, and going all the way down to almost central Illinois. So there's a lot of gradient change, latitudinal change. You'll see streams changing from cool water streams to like these warm ag ditches that we have in central Illinois. So I wanted to break up the community composition metrics just to see whether we see a difference. So this is the lower tributaries. So this is arbitrary. I just decided halfway between, I'm going to make this the lower part. There's no real biological reason for that. So I'm looking at the lower watershed here. We see the same 5 species about. Plain pocketbook, very common. Giant floaters, white heel splitters, ellipse, and fat mucket. So fat mucket is related really closely to the plain pocketbook. They're both yellow mussels and common, see them everywhere. When we look at number of individuals, same things, see a lot of fat muckets, ellipse, plain pocketbook, Wabash pigtoe, and round pigtoe. So this is the Fox River community. When we look at the upper tributaries, we're seeing same things. I was like hoping that we'd see something a little bit different, but really plain pocketbook, every site that you're going to find mussels at in the Fox River, you'll probably find a plain pocketbook. Creeper are really common, round pigtoe, Wabash pigtoe, and mucket. So mucket is one of those that is kind of more of a northern species. We see it in like the Kishwaukee watershed, which isn't part of the Fox, but it's the absolute most common mussel in the Kishwaukee and the rock systems. And it seems to hold the case too for the Fox River watershed, the upper part. Ellipse is our little shining star. We see ellipse lots of places. It's very, very abundant. Over 1,000 individuals collected in the upper tributaries. A lot of muckets. And one thing that's really encouraging to see is spike. So spike is a state endangered mussel in Illinois, but is stable in Wisconsin. And this number here is driven by one really spectacular site from the Mukwonago River, where they collected over 200 individuals. So just take this spike with a grain of salt. We have spike, I believe, at 3 sites that we know of in Illinois in the Fox tributaries. So we're hoping that it's hanging on in the main stem. We've seen shell records, but we haven't found it alive. We also haven't sampled much in the main stem, but it is doing well in a few of the tributaries in Illinois, not as well as it is in the Mukwonago. So anyway, common community seen throughout the entire watershed. Okay, but what we really wanted to know is where are sites that are doing really well? So I made a map here of the whole watershed, and you can see sites that had, or the number of species that are at each site. So blue here is good, blue is life. Green is still alive, but not as good as blue, I guess. And then the little black dots are places where we may not have found any mussels, or we found 2 or less species. And then yellow is 3 to 5 live species. So we're only looking at live species here. The good and bad thing about freshwater mussels is that they leave their shells behind when they die. We can take some information from that, in that if the shell is present, it probably was present nearby in the watershed, but we also know that shells can travel for tens of miles downriver, and then you might pick them up and think, well, maybe it exists at this site. We also know that shells get moved around by people. Fishermen will use them as bait. They'll put them in their boat just like they do fish, and they might end up in a different place. So for all of this information that I've been presenting to you, we're only looking at live individuals. We do keep the shell records. We have them. They're certainly helpful, but what we're really interested in is what is still alive. So we see 13 sites here in the watershed that have 10 or more live species, and they're distributed throughout the basin, which is really great news. Here you can see some of our hotspots in the lower part of the watershed. So this little circle here on the bottom left is circling Indian and Little Indian creeks. These are places that have very, very abundant and diverse populations of freshwater mussels. They are doing really well. Blackberry Creek is another one that's doing really well. And so these would be really great candidates for further augmentation of things like rainbow mussel that we don't have in the basin anymore, but we would like to see again. The other place in Illinois that had great diversity is the Nippersink Creek Basin. Nippersink is an amazing system to work in. The mussels are doing well there. It's also the last place that we believe or rainbow mussel were present in Illinois. They might still be hanging on there somewhere, hopefully, but those are some really great spots throughout the watershed. In Wisconsin, Honey Creek, the Mugwanago River, and the Fox Main Stem, the upper Fox Main Stem in Wisconsin, had sites with more than 10 live species. So all of this is really, really encouraging. We're seeing sites throughout that are really diverse, and the only sites that are kind of the most impacted are these in the middle, which are also the ones that are closest to the urban center of Chicago and these outer suburb areas. But even there, we're still seeing mussel populations, nearing 10 species, which is really good. And some of these dots that maybe didn't have 10 or more live species, they do have populations of state endangered mussels. So Fersen Creek, Otter Creek, those are places that have spike populations and are doing okay, but they just don't have quite as much diversity maybe as these other sites. Okay, so we just started looking at these data maybe a month or two ago because we all got everything compiled. So there will be more analyses to come. This project will have a large report that will be produced that I'm sure will be distributed to all the people. So if you want more, there will be more. Okay, so some species highlights here. This is ellipse. Ellipse are a very small bodied mussel. They only get to be 2 to 3 inches long. There's a couple big honkers that we get that are like maybe 3.5 inches, but in general, they're very small. They're also hosted by small-bodied fishes. Like there's a few species of darters that will host ellipse, as well as a couple species of sculpins. Sculpins are a fish that typically inhabits clear, cool, flowing streams with rocky substrates, which is exactly where ellipse love to live. Ellipse are really interesting during their reproductive period because they just they produce a tiny little lure to attract the fish, and the females will just kind of like pop out and lay on their side and just sort of wait. So they're targeting these small-bodied fishes like darters and sculpin that are coming along. They live on the bottom of the stream, they're looking for food, and they happen upon this weird looking thing that's like this tiny little lure inside the ellipse, and that's how they make that connection. So ellipse in the Fox River system are really, really common. Like I said, we found thousands of individuals of ellipse, and it's great to see because they do kind of inhabit these streams that have rocky substrates. Streams with bedrock sometimes will still have ellipse, and it's good to see. They are listed as threatened in Wisconsin, despite how many individuals were found. I don't know whether there will be updates to that, but I believe they have kind of a restricted range. And they are listed as conservation concern in Illinois, and this is also just due to range. They're mostly found only in the northern part of the state, but where they're found, they're doing quite well. They were among the most abundant mussels that were collected. One of the things that we've learned, or that we know, I guess, about ellipse is that they look really similar to rainbow, which was the target of this project for the most part. So this picture, this is an ellipse, and this one is a rainbow. And this picture actually is from a site that fooled me at one point because there were so many ellipse that was the only thing on our mind. We just thought we were looking at ellipse. But this rainbow came from a watershed that's a direct tributary to the Illinois River. It actually flows into Oxable Creek, which flows into the Illinois River. And there's a population of rainbow that is in existence there. Was not known to science before 2017. I don't know whether it had been mis-ID'd as an ellipse for so long, or if it just happens to be in that system and the system was never surveyed. It's hard to know. Either way, when you're working in a watershed that has lots of ellipse, but you're also looking for a really specific mussel like the rainbow mussel, this is challenging. Even for malacologists that are well trained, we have a hard time distinguishing between the two species. When you have the shells that are open and they're dead animals, it's a lot easier to see because ellipse have really heavy teeth, the internal teeth structure, and rainbow have thinner teeth. So, but if it's alive, because these are both species we want to keep alive, it's hard to distinguish between them. So this is our rainbow mussel. Rainbow was a really huge focus for this project because they are state endangered in Illinois and Wisconsin. Prior to 5 years ago, we believed there was only one population of rainbow in Illinois, and that's in the Vermilion and the Wabash drainage, which is hundreds and hundreds of 3 miles away from the Fox River Basin. But we discovered a population near Morris, Illinois, in a small tributary system to the Illinois River. And then there are a few populations that persist in Wisconsin. They're present in the Mukwonago Basin, the East Branch, Milwaukee, and I believe the White River system. So there's only a few populations remaining in between Wisconsin and Illinois. And this is a species that actually seems to be doing okay in places where it exists. In the Vermilion Basin, it's really common in some of the sites along the Salt Fork Vermilion River. It can live in agriculturally impacted systems. The system that it's present in, that's a tributary to the Illinois River, is a really, really impacted system. I mean, it's a series of drainage ditches basically, and they're persisting there. So it's a species that we believe we have really good chances of being able to propagate and augment populations with success. They are hosted by smallmouth bass, largemouth bass, and a couple darter species. So I'm going to show you. Okay, here's a video. This is a video of from Missouri, a little creek in Missouri, and rainbow will make a crayfish lure, which is why smallmouth bass and largemouth bass are really attracted to it. Let me go back and see if I can play it again for you. There we go. So it actually made little claws, if you will, and it's clapping them together while it also is swinging its body through the rocks to make it look like it's a crayfish that's moving. I mean, it's crazy what these mussels can do without having eyeballs or a brain. So it's really cool. So we know that they're hosted by smallmouth bass, largemouth bass, rainbow darters, yellow perch, fish that are super common. So lots of times in mussel conservation, the issue might be a lack of host fish. But for rainbow, this is not the case. The host fish are present at every single site. I think they're also hosted by green sunfish, so not a problem. There's green sunfish everywhere. So we're excited about the prospects for this project. Okay, I think I said everything on this. Yeah, we're hoping to work towards augmentation in both states. I'll talk more about that at the very end. But I just wanted to show you the kind of difference between these two species. One is the ellipse, very, very common, very prolific, really similar in size and shape to rainbow. So if you're working in these watersheds and you think that you might have a rainbow, that's great. Take pictures if you can. Pictures are really, really helpful, especially if we can get a picture of the mussel, the profile of the mussel, as well as a close-up of this little section at the top, like pointing towards us. I should have put a picture on here of that. That's the beak. The beak sculpture is really key for a lot of species of mussels. So if you're going to email pictures to a mussel expert, try to get a really nice in focus picture of the shell and the beak, and then we can help you out. If you have a shell that's open like this one, that's also really helpful. And we're happy to take shells, especially if it's something really interesting or rare or a new record of something. Those will probably be cataloged and deposited into the museum at INHS. So anyway, I'll just put that out there. Okay, crayfish lure, we already saw it. This video came from the Unio Gallery. If you love freshwater mussels and you want to watch more videos about freshwater mussels, you can go to the Unio Gallery, just Google Unio Gallery, and you'll find lots of pictures and videos, and it's really amazing. Okay, so I promised I would talk more about this. This is the study that was completed in 2004 by Bob Schanzel, Glenn Cruz, Joe Kath, Roger Klosek, and Kevin Cummings, and they surveyed the entire watershed, like we have just done as well. 96 sites. They did sample the main stem quite a bit, and in that study, they found 23 live species, and we found 24 live species, which I think is really exciting. The species that we collected that they did not collect in the late 90s was pink heel splitter. So Botamelus elatus, it was found alive at a couple sites in Wisconsin, which is great. All the other species matched. That's the end of the story there. They found high quality sites in the Mugwanago River, of course, and also in the Fox River in West Dundee, Illinois. We sampled the fox in West Dundee 2 summers ago, 2021, and it is a mussel bed. It's awesome. I have not seen that many mussels in a place in the Fox River anywhere else. We didn't collect as many live species there, but they could be some, individuals that were just not present at the time when we were there, or they eluded us. But anyway, those 2 high quality sites are still high quality sites now. The most common species that they found back then are the same ones that we have been finding now: plain pocketbook, pimpleback, giant floater, and white heel splitter. So these are your mussels of the fox. So we are looking a little bit more in depth at this comparison 25 years later. So I mentioned that it's a reasonable time frame to think about changes in the community. So we don't see in freshwater mussels change very quickly because mussels are sedentary. They can't swim like fish do. They can't move 300 miles like you might see a freshwater drum doing, but they do move mostly downstream, and the juveniles will move, or the larvae will move on fish. But it still takes a really long time to see any kind of changes or recoveries happen. So 25 years seems like a reasonable time frame to look at changes in community or changes in diversity, that sort of thing. So between our 2 studies, we have 46 sites where we sampled nearly the exact same location. We're using a buffer of about a half mile to look at comparisons. So first I want to show you richness comparisons. This map is showing changes in richness. So blue means that we found more than 3 more species during our present day study in 2021, 2022 than were found around the year 2000. Brown means we didn't find as many species, we found fewer species, and green we're saying is no change. There might have been a change in 1 or 2 species, but really not much change. So I do want to point out that there are sampling biases with everything. So it's possible that one of those samples that was reported by the Schanzel study, it was a bad day for surveying. It might have been raining or cloudy or turbid, or they just, parked on the wrong gravel bar, and then we happened upon the right one or whatever. So saying that we've found 7 more live species doesn't necessarily mean that stream grew 7 species in the last 20 years, but it's possible. I don't know. Anyway, so I just want to point that out. We know that this isn't like perfect 1-to-1. This is more just looking at a trend for the basin. So many of the sites had at least more than 3 more species. So that's really incredible to me. 16 of the sites had more species than were found. The average species gain across the whole watershed comparison was about 1 species, but that's at least something that's going in the right direction. Seeing species gains is awesome to me. Okay, most of the sites were about the same. 25 sites had no real change, and a few sites we did find fewer species during our survey than were found 20 some years ago. And so the change ranged between a loss of 5 species and a gain of 7. These are just basic algebraic comparisons. We are working on looking at similarity and dissimilarity coefficients to look at whether the community is the same, whether we're seeing like an actual shift in community, because we do see that happen in some populations where the host fish may be different now, and you see a slow change in actual community structure. And we're relating that to some predictor variables that we have in the watershed, like distance to the main stem or amount of land use change from ag to urban, or maybe ag to green space, or that sort of thing. So we just started looking at this, and I just haven't had time to compile it yet. When you look at abundance, you see we're finding more individuals than were found in the previous survey. Some of this could be, I will say that we compared sites for 4 collector hours from our study and the Schanzel study. So we're trying to compare effort, but it's really hard to correct for effort. If you have 5 people sampling for 45 minutes, it's not the same as one person sampling for 6 hours. It's just not. But anyway, it's the best that we can do with older data, and that's what we're doing. So in general, we were also seeing more mussels at all the sites, which is great. 16 sites had more mussels than 25 years ago. And I used a really generous distribution here. So if there were more or less than 50, I called that no change. More than 50 individuals collected, I called that more. So at almost all the sites where we saw more species, we also are seeing more individuals. Mussels in general like to be with other mussels. So they're patchy in distribution. Because of hydraulic changes in the river, mussels will end up in one site just because of how the river will sort itself out during high flows. So when we see mussels, we usually will find more mussels. If you're at a place that has no mussels, then there's probably not going to be that many mussels. So we're seeing an increase in individuals as well as an increase in species throughout the watershed in these sites that are doing well. So you can see, I mean, the sites that we found high species richness, like Indian Creek and Little Indian Creek, Blackberry Creek, Mugwanago, Honey Creek, those places, they are improving. And so that's a really encouraging sign. I know I was doom and gloom about the Fox River main stem. I'm not going to go back on that because we still have a ton of work to do in the Fox River main stem to get populations even persisting above like 8 live species in most places. But these tributary watersheds are doing well. It's hard to really pinpoint how and why, but they are. And so I'm hopeful that these will be source populations to eventually get back to the Fox River to boost those populations. Again, the Fox River is impacted by dams throughout the entire length of its watershed. So we might be able to improve populations in a few places on the main stem, but without some like major intervention, there will never be continuity between populations on the Fox main stem. Okay, so next steps here, more work on this comparison analysis. We just started looking at it and are really encouraged. We are trying to step back in time 25 years from the 2004 study to see whether we're seeing similar trends and to actually look at these changes in community. The data, like I said, start to fall apart before present day, I guess. We just weren't taking as rigorous data or they're not available to us, but we're looking at these where we can. So the work that is happening right now is the augmentation of rainbow in Wisconsin. So Genoa Fish Hatchery is working on growing the broodstock, getting the reproductive cycle happening for the rainbow that were collected, and those will be used to reintroduce or I guess boost the populations that are existing right now in the Fox River in Wisconsin, in the Mugwanago River. So broodstock is being taken from the Mugwanago and then put back, basically just kind of boost these numbers. Because like I said, mussels like to have more mussels. And so these will hopefully make these populations a little bit more robust if there were any kind of stochastic event that impacts the species. Okay, so other work that's happening in Wisconsin is some further work with fish interactions. Todd Levine at Carroll College is working, I think, in the White River, maybe, or no, in the Mugwanago, to do some more research on actual fish interactions with rainbow mussel. So rainbow will host on a lot of different species, but all that knowledge is based on lab interactions. So we don't really know what happens out in the river. And so he's going to be using some underwater photography to look at that sort of thing. Okay, we would love to work on augmentation in Illinois, working towards similar goals. So we have started the process, working towards the SSA, the Species Status Assessment in Illinois, which is required to do any kind of recovery work for any species. So we're starting that process, hoping to get to the point where we can actually augment populations in Illinois. Part of all of this is a genetic analysis that we've been working on with Ken Inouye that's at the Shed Aquarium. So we've been taking, throughout this project and other projects, we've been taking genetic samples of rainbow from all the populations that we have in the upper Midwest. So we've been taking samples from our Wabash population, the population in the Illinois River, the few that are in the Fox, and those in the Milwaukee River, I believe, in Wisconsin. And so we're looking at the genetic structure. It's really important with freshwater mussels because there's some cryptic diversity that exists out there, that we make sure that we're doing the best that we can for making sure that we keep the right genetic balance. The really preliminary results from Ken's work is that this is all one huge population of rainbow, which is really good news. So hopefully that means we can draw from broodstock from other places. Okay. I want to acknowledge everyone. I know that I missed some people, but we have really been so pleased with all of the county conservation districts that have been helping us, getting site access, providing data to us. We have a lot of data from county conservation districts, but if you want your data to be used in this kind of bigger picture analysis, and you don't know if we have it, feel free to contact me, which I didn't put my contact information up here. So I will put that in the chat box. Or I can put it on a slide here after this presentation's over. So thank you so much for everyone, just for your interest and desire in freshwater mussels. It's really helpful for people like me that work with a group of animals that most people have never even heard of. So I can take questions. I'm happy to talk more about any of the things. I'm going to put my email up so everybody can see it. Or my phone number, or whatever. I don't know. Are there questions?
Yeah. This is Jim Anderson. I'm working with Citizens for Conservation, and I'm wondering, have you visited Spring Creek and Flint Creek? And what was your impression of those two streams? Or are you going to be doing more sampling? And can we help you with that on those two tributaries?
Okay, Spring Creek. And what's the other one?
Flint Creek.
Flint.
Right.
We did go to Spring. Now, is this the spring that goes through Fox River Grove?
Yes.
Okay, we worked on Spring this summer, and it was really neat. I have a table. I'd have to look at the actual table to tell you exactly what we found, but we actually found a live spike in Spring Creek at Fox River Grove, which was really cool. We found a pretty diverse population there. It wasn't like overly remarkable, but it's a really small stream. But it was great to see spike. We saw lots of creepers, and I can look at the data. If you want to send me an email, I can share the data with you from Spring. Flint, I do not remember. I believe we sampled there, but it doesn't like stick out as a place that was like remarkable or unremarkable.
Can I ask you, Alison, also, did you get into headwater streams when you were sampling, or?
So we did sample some headwaters. So the way that we determined our site list was, we looked at the previous study to make sure that we kind of visited similar sites or sites that were nearby. I also was using data that I had worked on maybe 10 years ago, looking at pre-colonization estimates of where rainbow might be present, because we were really targeting, we're interested in targeting rainbow for this project. It ended up that we got data from the whole watershed for all the species, but we really were interested in finding out if rainbow were still present anywhere in the Illinois part of the Fox River and where else it persisted in Wisconsin. So I use these pre-colonization estimates that use, we use maximum entropy to kind of predict where rainbow might or probably were present hundreds of years ago. And we tried to match places where we didn't have great data for to pick new sites. So we did that, and also matched up museum shell records, which mostly are on the main stem. I do not think, this is my professional, also personal opinion, I don't think that there's still rainbow in the main stem Fox. I think the shells will wash down the tributaries and end up there, and that's where we collect them. But either way, we were trying to pick places on tributaries that were close to shell records that we have. So we picked a few sites just by looking at maps and determining like, well, this could be a remnant population of rainbow that we don't know about. And for the most part, those small tributaries or really, really high up headwater streams did not have freshwater mussels, which is expected. We don't expect there to be like remarkable populations in those really small streams. So yeah, we try to get coverage of the whole watershed. There are places I'm sure we missed. If anybody wants a list of the places we've been and then maybe has a place that we haven't been that you're like, oh, this is awesome, we have mussels here, we'll come and sample it for sure. Like I would love to know those places.
Great, thanks.
Hey, Alison, this is Cindy Skirkrud.
Hi, Cindy.
Hey, how are you?
Good.
Thanks for your help. I know that in Wisconsin, because of their rules, there was this citizen scientist effort that Jesse and Lisa, train people to go out and sample. And I just wondered, is there any, like, did that help at all to find more rainbows or?
Yeah, that's a great question. Are you talking about river keepers?
Yeah.
I think it's river keepers. Yes, I know they worked with river keepers. I don't know much about Milwaukee River Keeper. I don't know much about the results from that, but I know that they said they had really amazing turnout and a ton of volunteers that would help with that. But I can ask and find out whether, I guess, how fruitful it is to have those smaller watershed groups. I will say, unrelated to the Fox River Basin, we had a similar turnout for River Watch, which is a program through NGREC in Illinois. And they're kind of piloting a freshwater mussel program through River Watch in a few different basins. And one of the sites this summer on the Sangamon River, which is a central Illinois stream, they found the first spike ever to be found in the last 40, 30 years, 35 years, hadn't been found alive. We just assumed it was gone. And because they had so many volunteers out there and so many hands, they were able to turn up a really rare species, which is great. So lots of times those, what seems, just to be like a feel-good exercise with volunteers, they actually do turn up really useful information, because when we're out there with a crew of 3 or 4, it's hard for us to get in all the places. And so having larger sets of people is helpful. I mean, it can be harmful, I guess, if you're targeting the same mussel bed over and over and over again. That's something we don't love to see, but in general, I think it's great. And the more people that get to be out in the rivers and finding freshwater mussels, the better.
Great, thanks. Thanks for that info.
I had a quick question for mussel propagation. Have you looked into using, I know DuPage County has a full wet lab that they've been
Yeah, we've met with them a couple times. I know they're doing some really good work in the DuPage system for sure. We are in contact with them. They're actually growing some mussels for a different project that we need little bitty juveniles for, and we would love to partner with them. Illinois DNR has restrictions, or maybe we just haven't gotten there yet, for propagating state listed animals for reintroduction. I'm hopeful that we'll be able to use that as a conservation tool in the really near future. But right now, Illinois is not propagating state endangered mussels. So, but yeah, DuPage County would be a great facility. They are really equipped to do this exact thing. It would be a logical place to have an operation. Okay, and Jeff said they'll be presenting at the summit as well. And yeah, so Lisi and Jesse, or at least Jesse and I believe Todd, will be presenting at the summit, which is in March. Is that correct? And so they'll be presenting probably a lot of the same stuff I may have already talked about, but also some additional information there too.
Yeah, how much longer will the field work be going on?
How much longer will the field work be going on is the question.
Yes. So we have, okay, March 16th is the Fox River Summit. We pretty much sampled most of the streams. So there were a few streams on the Illinois side that we were unable to get to this year because of reasons. And so we will be sampling a few sites this year. We have a partner project with DNR looking at quantifying change after construction events. And some of those sites overlap sites that we need to sample for this project. So we'll be in maybe 8 to 10 sites again this coming year. But I believe after that, the fieldwork component for actual sampling or just surveying will be done. But the propagation effort will continue on the Wisconsin side for at least this season and I think next. But again, if you have a place that you think might be a hidden gem that we don't know about, please feel free to reach out and let us know, because that's the thing. I mean, I'm based in Champaign. I'm 3.5 hours away, and Wisconsin DNR are in Madison and places north. And so you all are really like our hands and feet and eyes and ears, and we're just happy to always hear from people that are much more knowledgeable about the system.
Do you look at fingernail clams as well?
Sure. Yeah, fingernail clams are hard to identify for me. There are a few experts in the field that can distinguish between them. We will collect fingernail clams, pea clams, as well as aquatic gastropods. That's kind of my side hobby, is looking at freshwater snail diversity. It's not a focus for this particular project, but we did collect all those types of things. For fingernail clams, if you're not familiar with them, they are native clams, not to be confused with corbicula, which are an invasive clam. But our native clams, we probably have 30-ish species, I think, but I don't know how to identify them. We sent a few to a grad student to identify, and he sent them back with names. And so those are the data that we have, but I'll put a genus on it, and that's all that I can do, really. But yeah, fingernail clams are one of those like unknown components of diversity, which is the same for snails. Snails are believed to follow the same pattern as freshwater mussels do as far as impairment. And a lot of them are difficult to ID. A lot of our snails are only a few millimeters in size. You have to identify them under magnification. Their characters are really different based on the type of water that they're in. And so they're hard to identify. So we do collect them, both pea clams, fingernail clams, and snails, but they're hard to ID. The other thing that I wanted to look at, which we haven't had time to look at much, is invasive species. So there's at least 3, no, there's probably more, invasive mollusks that are in the basin. Zebra mussel, quagga are probably present. Corbicula, there's at least 2 species of corbicula, as well as the mystery snails, Chinese and Japanese mystery snail, which are really large snails. If you see those in a stream, if you see a huge snail in a stream, it's probably an invasive species. And then the other snail that we're seeing pop up are faucet snails. So we're seeing those more and more commonly. They're small, so they will go undetected to most people. So anyway, we're trying to at least keep our finger on the pulse of where these invasives are and where they're spreading to. In general, they're not too much of a problem, I guess. I don't know.
I have a question. Alison, your data suggests that mussels are doing pretty well, increasing in abundance in some sites and maintaining their own populations, increasing in species richness. Are there changes in the landscape that you attribute this to? Changing agricultural practices, improved water quality? Have you looked at some of those features of the landscape that could explain how well the mussels seem to be doing?
Yeah, that's what we're hoping to actually do a statistical analysis to look at. Obviously, the main thing that has changed is the Clean Water Act, but it's been 53 years since the Clean Water Act started. But because mussels are long-lived and it takes a long time to see these changes, I believe that we're still seeing positive impacts based on that, landmark decision 50 years ago. I also think some of it is sampling bias, that we are more effective at sampling because we had a really well-funded project. And so we could have staff to be in the rivers at the exact right time to be sampling. So that's something that you can't ignore, that we just had a lot of resources and time to do this project well. The surveys in the late 90s were really well done. And so it's really hard to tease apart whether this is like a sampling bias or actual improvement in the community. I also think that improvements in water quality are helping. I don't want to say, I can't say right now what the smoking gun is of, you know, what the cause of having more species and more individuals is. These large populations of freshwater mussels sometimes also isn't like a great thing. I've been working with Wendell Hagg, who's a really well-known mussel researcher that works for the Forest Service, and he's working on some of these hypotheses that mussel populations that are quick growing, like fast growing species that go from being really little to really big really quickly because they're in warmer water than typical, they might be more likely to go through this boom bust, you know, so you have a lot of individuals. Hopefully they can reproduce before they get killed off by whatever stressor happens. And we are seeing increased water temperatures throughout everywhere because of climate change and reduced forestation and all those sorts of things. And so it's a delicate balance. I'm hoping that this is a positive trend, but we'll just have to see. So sorry, I didn't answer your question. I don't know. That's what I mean to say. I'm not sure.
Other questions?
Alison, this is Nancy with Fox River Ecosystem Partnership. Will you be able to make this available for posting on our downstream website, this presentation?
Yeah, I can make a smaller version so it'll be easier to download or host or whatever. And I know it's been recorded. I don't know where that will be, but maybe someone else can answer that.
Yeah, the video will be posted and shared.
Okay, great. Thanks.
But yeah, Jeff, I can send you like a PDF of the slides or the actual presentation or whatever you might want. Okay. Yeah, I'm happy to have it be on the webpage. That's fine.
Okay, thank you.
Are slipper shells holding on, or are they?
No, there are slipper shell. Some of the streams we found quite a few slipper shell. They were on the state endangered list in Illinois up until the last revision, I want to say 2015. They were believed to be a rare species, but so the project that I started on when I moved to Illinois was doing a statewide mussel survey. So myself, Sarah Douglas, and Diane Chastain, we worked through the entire state, sampling a lot of streams, and those data helped explain that slipper shell actually are not that rare. They're just a small headwater specialist. So we did find a few sites with a lot of slipper shell, or, you know, a normal number of slipper shell. So they're actually doing okay.
All right, looks like we may have exhausted all of our questions. So
Whew.
thank you, Alison. We really appreciate your presence here today.
Yeah, you're welcome.
You're sharing. Thank you for all the hard work you're doing as well. You know, thank you.
That's the fun part. So yeah, and feel free if you have additional questions to send me an email. So I will stop sharing now. Do you need me to stay on the line, or you guys are gonna have your meeting?
I think we're good. We've got a couple of member announcements, but yeah.
And isn't Cindy gonna talk?
No, there's nothing for that.
Oh,
Alison, you are a fabulous presenter.
yes.
Thank you. I know it's not as fun not being in real life, but hopefully it was enjoyable for everyone.
It was real enough. Thank you for your time. So I guess you're excused if that's.
Okay, thank you.